Welcome to Eurofins Viracor Clinical Diagnostics Podcast, where I'll be sharing up-to-date scientific advances, key diagnostic testing, and our most recent innovations. Welcome to our podcast. This is our fourth year hosting our live in-booth interview show, Transplant Radio. Our aim is to connect with the transplant community, a diverse group that includes professionals, donors, patients, and even doctors who are also recipients. If you'd like to be a guest on our show, you can find our email in the episode summary. Amplify your influence on Transplant Radio. Enjoy episode 1 of our 15-part series.
Welcome to Amplify Your Influence at the World Transplant Congress here in sunny San Francisco, California. It is Saturday, August the 2nd, 2025. I'm sitting here with Mr. Evan Sander. Evan is a well-seasoned, well-respected R&D scientist with Eurofins Viracor, located in lovely Lenexa, Kansas. Go ahead and introduce yourself to the audience, Evan, and tell us a little bit about yourself and what projects you've been working on lately. What brings you to the WTC 2025?
Yeah, it was my first time at WTC, here to present a poster and abstract that will be published in the American Journal of Transplantation tomorrow, and it will be for a new qPCR assay for Chagas disease that detects nucleic acid from the protozoan Trypanosoma cruzi.
All right, so give us a little bit of background about Chagas disease. The bug also has a very interesting nickname of the kissing bug, or not to be confused with the kissing disease, which we all used to say was what caused all the mumps outbreaks back when I was a kid. I don't know about you, but they probably changed it to something cooler since then. So tell us a little bit about, how the organism, the T-qPCR I don't want to say parasite, but that's really what it is, becomes infected with the agent and then transmits that to the human population, and a little bit about the life cycle within the human population, and maybe some signs and symptomology of what people can look for to say, hey, maybe this is what I've got, this is why I'm not feeling so hot today.
Sure. Yeah, Trypanosoma cruzi, or T-qPCR, is endemic in South America as well as Central America, and the main life cycle is that it lives within the gut of the triadenine vectors, which are also referred to as kissing bugs. And those, especially in rural environments, can bite people, and when they bite, and then they will basically defecate in the wound, and then you itch it into your skin, then that is the way that the parasite is then transmitted into the blood. There's a different life cycle for this protozoan where it exists as a tryptomastogote when it's in the blood, and the tryptomastogote can then travel and then become locate itself in different organs. Usually the heart is the main organ that it likes to go to, but it can also go to the intestines and go into the cells there. And once it gets into the heart, for example, then it converts into a more replicative stage called the amastogote, and then the amastogote will then replicate and stay inside the heart.
All right, for everybody that doesn't have their WTC secret decoder ring, you said tryptomastogote and amastogote are not a goat, not the greatest of all time, that goat, but explain a little bit about the difference between those two and exactly what they mean talk to me like I'm a fifth grader, because that's my wife says my mentality is most times.
Sure. Yeah, so they're just different life cycle forms of the same single cellular organism. And so the amastogote, they're a little bit smaller. They don't have the tails, which we call flagella, so they don't swim around. They're just there to replicate and divide. And then the tryptomastogote is a larger, more aggressive form. If you look at it under the microscope, it's a little bit bigger and it swims around a little faster. It has a tail. And so that's the point, that it's not replicating, it's not worrying about growing, it's just about getting out there and infecting new people or getting back into the triadenine. And then there's another one that I didn't mention called the epimastigote. And the epimastigote is actually what we use in the lab, because that's what, when you culture them, they exist in the epimastigote stage, and that's the same stage that exists inside the gut of the triadenines. And so those also have the tail, they also swim around, but they're a little bit smaller and they're not quite as aggressive as the trypanomastigotes.
Okay. And you had said something earlier that it goes through the blood system of the host, the person, but then it tends to reside in heart tissue and such. So it doesn't really infect or get into the blood cells. It goes more to the myocardium or to the heart muscle tissues.
Exactly, yeah.
And such. And what happens once it sets up a new residence then in the heart tissue?
Yeah, and so that's kind of what makes it relevant to transplant medicine, because when you first get infected, that's called the acute phase, the infection, and there's usually some symptoms, but it's kind of vague, just like anytime you get sick, you're a little bit tired, stuff like that. But then eventually you go into the asymptomatic stage called chronic Chagas disease, and there's a lot of people that are immigrants, for example, that live in the U.S. From South America or Central America that have the chronic stage of disease. You wouldn't know they have it because they're completely asymptomatic, but they have it inside their heart or maybe some of their other organs. And the only way that you know if they have it is through a serology test, where you look for the antibodies because they've been infected. They should have an immune response, and there are tests available in the U.S. and elsewhere that look for the antibodies.
Okay. So typically what you would look for, if it's early stage in the infection, you look for an IgM marker in that. If it's a little latent stage or later stage, you go for an IgG type response in that. But aside from doing serological markers, what is the particular marker? You said a qPCR assay that's being used by Eurofins Viracor to help identify the presence of this organism in the human body. Explain that a little bit.
Yeah, so qPCR is able to detect in the blood. So if you get a blood sample and you extract it out, then if there's any trypanomastigotes that are in that tube of blood, we can get the DNA and we can use qPCR to amplify it, detect those there. The reason why that's important is during an acute phase infection, you will have trypanomastigotes in the blood, so you can use it for that. But what we're mainly concerned about is if a patient who's getting a transplant receives an organ from someone who has chronic Chagas disease, there's a risk that about 30% of the time, or greater if it's a heart transplant, they can have a reactivation event, where that, because they're heavily immunosuppressed, the trypanosome will go into their blood and can cause catastrophic damage if their immune system can't fight off that infection.
And so it's like some of the other latent viruses and like that in the body. The body mounts a response, it knocks down the total count of whatever that infective agent is, but it's still hiding out in the body somewhere. You become immunocompromised, your immune system is impacted some way, either through therapeutic agents or aging or other disease factors that cause your immune system to decline. So then they're like, oh, here's my opportunity again. They'll come back up and cause more problems or be more not really visible but detectable in that patient's blood sample. So you're presenting a paper here at the WTC Conference 2025 tomorrow afternoon, I believe it is.
On Tuesday.
So a couple of days.
Mm-hmm.
Great. Why don't you kind of walk us through what your paper is about, what your hypothesis was, your methodology, and then what a discussion with the results that were obtainable and that you will demonstrate as you're going through your poster presentation on Tuesday.
Yeah. So the immediate need for this test is the fact that there's no commercial tests in the U.S. other than the test that we just launched that does a qPCR assay for Trypanosoma cruzi. And so if you're getting an organ from someone who either has confirmed chronic Chagas or they're from an endemic region and they might have it, then there's a need for a highly sensitive and very fast turnaround time test that is able to detect whether or not they have a reactivation event. Because not everyone will have a reactivation event, and if they do, there is highly effective drugs that can treat the infection. And so what you need is just to know whether or not it's happening. So our assay is actually a panel of qPCR assays assays that can detect different genetic targets within the organism. And so several of these, there's two components of our assay, which makes it a little bit unique as opposed to some other assays that are available in other parts of the world, like South America. And that is that there's a qualitative component, and then there's a true quantitative component. And I'll explain what that means in a second. But the first component is the canonical approach to amplifying this organism, which there's usually three targets that people use in the literature. The first is the kinetoplast DNA, and that is this very unique element of trypanosomes, which is basically a concatenated network of DNA mini-circles and maxi-circles, which is very similar to mitochondrial DNA. But there's a lot of unique structural components of this. It's a repetitive genetic element, and it's located near the flagellar motor. But the important thing is that there's tens of thousands, even hundreds of thousands of copies of this conserved genetic element in these parasites. So even if there's a single copy, if there's a single parasite in the blood sample that gets extracted, then we're having thousands of DNA molecules that we can amplify. So it's extremely sensitive. It's basically, you're not going to have any false positive or false negativity if you have this very high multi-copy target. The other ones that they use are satellite DNA, which is a nuclear target. So it's part of the nuclear chromosomes, because these are eukaryotes, so they have chromosomes just like people do. And this also exists in tens to even hundreds of thousands of copies, depending on the strain or the evolutionary lineage. And then finally, the 18S DNA as well for the ribosomes can be targeted. And that's also multi-copy, but usually more like tens or hundreds of copies. And so these are all really useful, high sensitivity targets. But the one thing you can't do with these targets is quantification. And the reason for that is that there are 6 major evolutionary lineages for this organism, and they're referred to as discrete typing units, which are almost different species even, but they're classified based on their DTU 1 through 6. And these all would have conserved kinetoplast DNA, satellite DNA, 18S. So those targets, it really is irrelevant what lineage they come from. But they have very vastly different numbers of those repetitive elements in their genome, depending on what evolutionary lineage they come from. DTU1, for example, is considered the evolution, one of the original evolutionary strains, one of the parent strains of some of the other hybrid organisms that have a high degree of heterogeneity in the genome or heterozygosity in the genome. For example, DTUs 5 and 6 are thought to be consequences of hybridization events that happened early in the Trypanosome's history. So if you're basically detecting satellite DNA copies in the blood, you're going to get some sort of number if you use a standard curve to quantify it. And that number could be hundreds of thousands, even millions, but you can't really convert that to parasites per milliliter of blood unless you know how many copies per cell you have. And if the numbers of copies in the cell can be different by tens or even hundreds of thousands, depending on which strain you're looking at, you can't use that to quantify directly. So what our assay does that's unique is it uses a new target that no one's used for PCR before, and that's called the TCS-C5D target. And this target it encodes for a sterile C5 desaturase protein, which is not really important, but it is a nuclear gene that exists in a single copy within the haploid genome. And because this is a diploid organism, we know it has exactly 2 copies per cell. So if we use PCR to get a CT value, and then we use a standard curve to get a gene copies per milliliter value, we can convert that to know exactly how many parasites per milliliter were in the sample of blood that we extracted. And so that's something that's new to this approach, and that's something we incorporate into our panel. So we have multiple reactions. One reaction is just multiplex for those highly sensitive, very specific targets. And then we also have a quantitative component, which can tell you exactly how many copies are in the blood.
You had said something earlier about DTUs, and just make sure I'm recalling my information on this. But didn't they start off originally just with DTU type 1, and over time that's evolved into 6 different DTU subunits, and there's some commonality and yet differentiation between those DTUs that allow you then to specifically target them to make the identification, a true identification of this organism. Can you explain that a little bit more for me?
Yeah, sure. Yeah, so this organism has diverged since it's been existing alongside humans, presumably since humans have been in South America. And the DT1 is considered the oldest strain. It also has the smallest genome of all of them, and that's something that we confirmed in our lab because we've cultured up multiple strains from all the different DTUs in our lab, and that then, of course, accumulates these differences when it goes through different hybridization events and things of that nature, as well as just mutations within the genome. Something that's interesting about this organism and trypanosomes in general is that it uses a combination of asexual and sexual reproduction. And some eukaryotic cells will only do one or the other, but these ones can do both. And it's not exactly understood under what conditions they do one or the other. It's generally thought that when you culture them in the lab, they're doing primarily asexual reproduction, and it's clonal in nature. But we also know that there are experiments that show definitively that they can reproduce sexually as well. And so some of these hybridization events are thought to come from that.
So you say it's more asexual in an in vivo or in vitro situation into a petri dish testing setting. So do you think there's something associated with the host factor that allows them to do sexual reproduction and then changing of their DTU types? Or what do you think it is? What does the literature show as to what determines whether that organism reproduces asexually or sexually?
Yeah, I think bottom line is it's not really known, but what I've read about it seems to suggest that they actually reproduce sexually more often than what we had originally assumed. So the traditional dogma, or commonly most understood, is that they reproduce clonally, and the reason for that is because you don't see mixing between the different DTUs. But this kind of goes to what I was saying at the beginning about those could be interpreted as being different species based on your species definition, because when you look at, for example, people have done experiments about putting antibiotic resistant markers into the same DTU of organism, and they're able to spread between the members of that population, showing that they are reproducing sexually. And so what's probably happening, in my opinion, is that they have some sort of selection, so they're only sexually reproducing within the same lineage, and for some reason they very rarely, if ever, produce viable offspring with the other DTUs. And so if you mix up, one DTU and another DTU in an in vitro environment, you don't see a lot of mixing between the two, but you do see mixing within them. And so it's, for some reason, it's just a very rare event that those two are able to hybridize from different lineages and then produce this new, lineage that then will accumulate its own mutations. And so that's why we see that these organisms have a very dynamic way of evolving that incorporates both asexual and sexual components. And when you look at them in the lab, depending on what you're looking at, it can sometimes seem like one or the other, but really it's like a mixture of the two.
Okay. So you had talked earlier, and again, you can tell me if I'm incorrect, or most people do, but you said the DTU type 1 is the oldest presentation in this organism, and it's evolved to these others. Is it also able to use the prevalence, use that a specific type of DTU to say this is probably where that organism or where that disease originated from in that individual? I know you said South America, but it's not just isolated to South America. There's two cruzi in other countries, but South America is probably the most endemic at this time. Is that some information that's useful or obtainable through this type of technology?
So you can definitely, and that's where the CCSE5D gene was originally used for. That's the papers that we read that let us know that was single target, and we confirmed that. They can do these type of genotyping to figure out what strain the parasite is, and that can be useful for determining where it comes from, because certain DTUs are more common in certain regions, and so people in South America are more likely to be infected with DTs 5 and 6, for example, whereas DTU4 is more common in Central America. In terms of transplantation setting, I'm not sure how useful that information is because they will know the place of origin of that person already, and the type of treatment that you give them is going to be the same no matter what. So really what's important is whether or not the parasite's actively replicating in their body or not, and that's the information that we give them with our test. I think for us, always the focus was inclusivity, making sure that regardless of what strain or DTU they were infected with, our test would give a result, a true positive result every single time if it's there. And that's what we were able to confirm because we grew up, not only did our actual build of the assay with the oligonucleotides, the primers and probes we used, were based on in silico data from all of the different DTUs, so it was fully inclusive, but then we also grew up all the stocks and strains that we were able to get our hands on from the American Tissue Culture Collective, and grew them up in the lab and then tested our assay with them to prove that it was, no matter what lineage it comes from, we're going to get the correct result.
Yeah, I mean, I can see, as you said, it's really, it's a test, it's most applicable in transplantation, presence or absence of T. cruzii in a potential organ to be transplanted into somebody else, but also thinking about the transplantation space, you not only have your transplant surgeons, but you also have your infectious control physicians, infectious disease physicians, and my experience is, epidemiology, that's kind of cool stuff. So that's, I guess why I was asking the question about being able to say, oh, this person became infected when they were whatever region of the world they were in. So you've talked a little bit about the assay, great information. You've talked about how you set it up and such. What were your findings? And let's talk sensitivity, specificity. Hard, fast numbers. How few organisms does the assay accurately detect? And and then the conclusion for your paper.
Yeah, so sensitivity, because we have this panel approach, we achieve extreme sensitivity with our multiplexed, multi-copy target portion of the assay. When we validated this, we have to use contrived samples. And so the data, like in our abstract, for example, reflects the stochastic nature of contriving samples, which is that when you are trying to go down to what this assay is capable of, which ultimately is, if there's a single parasite in the blood, the qPCR assay is going to pick it up because there's going to be tens of thousands of DNA molecules that are specific for our primary design, and it's going to be positive. But when you will make these samples, what we did is we cultured epimastigotes in our lab, and then we mixed it, spiked these into whole blood, and then diluted it down. When you dilute down to, for example, like one parasite per mil, there's a stochastic element of this where when you do a serial evolution, you're not going to get exactly one DNA in every single tube of blood. And so for that reason, when we did our validation, we were getting about like 4 to 9 parasites per mil detection, depending on the strain.
That's still pretty significant.
Yeah, that's very significant. And when we ended up doing like our cutoffs, so like based on what our lab will output to physicians, it's based on a CT cutoff rather than a quantified parasites per mil cutoff. And we allow for standard deviations on both sides of that CT. And so ultimately, I'd be very confident that if there is a parasite in the blood that gets extracted, it's going to be a positive result sent back to the client. In terms of like an analytical validation, we got down to like 4 parasites per mil, and that reflects the fact that some of your samples that you're testing that are supposed to be positives are actually negative because you can't really control for the parasites being in there or not.
Very good, very good. And then you also talked about quickness of turnaround time of results, and what is the targeted turnaround time for this assay?
Yeah, so on the website it says 24 hours. It's kind of like the guarantee. But
that's 24 hours from specimen receipt at the laboratory, not 24 hours from collection.
Yeah. I mean, if it takes a couple of days to get to the lab, then there's nothing we can do about that. But from the moment, we get the sample, usually more often than not, it's going to be within a single shift. And so the way Viracor works is we get samples in the morning, and there are staff that are ready to extract that sample as soon as it comes in and report it before they leave. And so usually they're leaving around 6 o'clock. And so, sometimes things happen, and like maybe the machine goes down or something like that, and it might take a little bit longer than that. But most of the time, within 8 to 10 hours of receipt, they're going to be getting the result back.
Which is very important, especially when we're talking about cardiac heart transplant transplantation.
Yep.
You want to be able to, retrieve the organ, then get it into the recipient as quickly as possible because the recipient's not that healthy at the time, and being able to put a good, viable, strong organ back into them is going to greatly improve their quality of life, get them on the right path to getting better. So, all right, anything else you'd like to add?
No, I'm excited to be here at WTC. I think I was really excited to work on this assay. I think it turned out really well. I learned a lot from working on it, and I think it fills a very clear need in transplant medicine, so I'm excited for it to be out there.
All right, well, great. Again, this is Kevin Foth with Eurofins Transplant. Amplify your influence with Evan Sander, the R&D scientist from Eurofins Viracor, located in Lenexa, Kansas. We're at booth 821 on the floor throughout the 2025 conference. And thank you, Evan. Greatly appreciate it.
Yep, thanks for having me.
Have a good conference and good luck with your presentation.
Thank you.
All right, thank you.
Make sure to subscribe to this podcast and follow us on social media to stay up to date on all the latest information. If you want to help spread the word, please give us a 5-star review and tell your friends and colleagues to subscribe too. We're available on Apple Podcasts, Spotify, Google, Podbean, and more. With over 40 years of diagnostic innovation and specialized expertise in infectious disease, pre- and post-transplant, allergy and immunology testing for immunocompromised and critical patients, Eurofins Viracor is committed to helping medical professionals, transplant teams, and reference labs get results faster when it matters most. Viracor is passionate about delivering value to our clients by providing timely, actionable information, never losing sight of the connection between the testing we perform and the patients you serve.